Hello, my name is Kivilcim Ozturk, and the title of my poster is Parsing within cell type heterogeneity using phenotypically supervised single-cell analysis. In this poster, we describe a protocol combining functional metrics with genomic data to elucidate drivers of within cell type heterogeneity via the phenotype to genotype link. This technique involves using fluorescence tagging to label and isolate cells grown in 3D culture, enabling high-throughput enrichment of phenotypically defined cell subpopulations by fluorescence-activated cell sorting. We then perform a validated phenotypically supervised single-cell analysis pipeline to reveal unique functional cell states, including genes and pathways that contribute to cellular heterogeneity and were undetectable by unsupervised analysis alone. MDA cells embedded in 3D type 1 collagen matrix take on at least two distinct collective phenotypes, which develop from single cells over the course of 7 days. The majority of cells, approximately 81%, formed collectively invasive cell structures, a morphology characterized by a low circularity index. A smaller subset of tumor cells, approximately 19%, did not invade, characterized by a high circularity index. Based on their morphology, we term the invasive structures as networks and the non-invasive structures as spheroids. Cells undergoing collective migration as invasive networks or non-invasive spheroids were labeled, sorted, and subjected to single-cell RNA-seq analysis, which suggested the presence of three cell states: network, spheroid, and intermediate. Differential expression analysis revealed that network cells were enriched for gene sets associated with proliferation relative to spheroid cells. Although phenotypically similar to spheroid cells, intermediate cells were distinguished from spheroids by genes involved in differentiation processes such as positive regulation of cell morphogenesis involved in differentiation and positive regulation of epithelial to mesenchymal transition. In contrast, intermediate cells were distinguished from network cells by processes relating to cell cycle. This supports that intermediate cells may be poised to transition out of their spheroid state and raises the possibility that certain genes relating to cell cycle processes may trigger the intermediate phenotype to switch into the more proliferative network phenotype. We identified gene expression patterns active in single cells using non-negative matrix factorization, where 3 of the identified patterns best explain the expression profiles of spheroid versus intermediate versus network phenotypes. In order to assess the progressive relationship between cell states, we performed a pseudotime analysis where the learned trajectories connect spheroid cells to network cells through the intermediate cells following cell cycle progression, and we investigated the expression progression of differentially expressed genes along the pseudotime trajectory. We automated these computational analyses in a gene pattern notebook to provide a user-friendly interface, making it easy to apply to any single-cell RNA-seq data. This protocol thus provides an end-to-end strategy to characterize functional within cell type heterogeneity associated with visual phenotype-based morphological traits. By implementing the procedures described in our protocol, one can readily use the approach of photoconversion to functionally isolate any phenotype that can be described by visual characteristics. While the end user will have to adapt analysis strategies to their specific contexts, we anticipate that the use of our downstream bioinformatic pipeline should enable the identification of potential biomarkers that regulate their system and inform further validation experiments. For complete details on the use and execution of this protocol, please refer to our papers listed under the references section. Thank you for listening.